Maltase Deficiency Association was formed in 1995 by Randall and Marilyn House.
My wife and I became involved in this effort 14 months ago when our daughter Tiffany Laurel was diagnosed with Pompei's disease. Overnight, of course, our lives changed.
Their first step was to identify those who were actively working in the field of Pompe disease that year.
It became very apparent to me that many in the medical community are totally unaware of this disease. Although her symptoms were numerous and persistent, nothing wrong, quote unquote, was ever found. When Tiffany was finally diagnosed at the Mayo Clinic last year, we were told that this disease is extremely rare, with only 50 to 100 reported cases. Based on our experience, I'm convinced that this is greatly underestimated. The purpose of this organization is to generate support for the scientific community so that development of enzyme and gene therapy for Pompei's disease can be realized. Over the past year, it has become evident to me that though there are many interested parties, there is a definite lack of communication between researchers in this field. You're all familiar with each other's work, but many of you never have met or even talked to one another. With this meeting, we are hoping to foster discussions which will hopefully lead to a cooperation amongst investigators. If this meeting does only that, we think it'll be a great success.
Through reading scientific papers on Pompe disease and reaching out to the authors of those papers, they learned that there was research being done at a variety of centers, including Erasmus University in Rotterdam, the Netherlands, Duke University, the NIH, NYU, among others.
From your research, it's very evident that enzyme replacement therapy is a viable option for the treatment of Pompei's disease. And we hope it's in the not too distant future. For my daughter's sake and the sake of the many patients that we have made contact with during the past year, we need to go forward with this project and bring the future within sight. I believe we're all aware of the progress that is being achieved towards development of a therapy for the treatment of Pompei's disease. And as you can tell by some of the titles of presentation, there are serious efforts being developed in the field of gene therapy as the next future step towards a cure of this terrible ailment. It is truly amazing how much has been accomplished during the past 1.5 years. And Marilyn and I wish to thank you for all that you are doing to speed this effort.
In 1998, the AMDA co-hosted a conference with the NIH, and again, it was obvious that we were getting closer and closer to a treatment becoming a reality. 1999 was a pivotal year for the Pompe community. The first ever clinical trials for an enzyme replacement therapy started. First in the Netherlands, using an enzyme derived from the milk of transgenic rabbits in January. Then in June, Duke University started a trial using an enzyme derived from a CHO cell line. Both of these trials were in infantile onset patients, but in June, the Dutch team also started a late onset arm of their trial.
The good news is that there are 2 drugs on the market that work. They're not on the market, excuse me. There are 2 drugs in trials that we know work. And Marilyn and I know firsthand because our daughter is receiving the drug in the Rotterdam trial.
It was at this conference in 2001 that patients heard firsthand the results from the trials that started in 1999. And the results were clear. enzyme replacement therapy for Pompe was no longer a dream or a hope.
We study structure and function of alpha-glucosidase in order to learn about this enzyme. We study structure and function of the gene in order to learn about gene function. We study correlation of genotype and phenotype so that by looking at the DNA we can give a diagnostic answer or a prognostic answer. Then of course we have studied how to produce alpha-glucosidase, and also we are studying how to apply and evaluate enzyme replacement therapy.
But proving it worked was just the first step. At this conference, the FDA spoke about the process of taking a therapy from the lab to the open market.
After the clinical investigation phase of the drug has already been completed, what happened is that the drug company would compile all of the data. They would make the analysis of the data to convince the FDA that the drug is actually safe and effective. An average NDA or BLA would contain approximately 100,000 to 160,000 pages. For us, for a team of approximately 5 or 6 people, to go through all of the information, all of the data, in order to, at the end of the day, we can say, is the drug safe and effective?
Very much like the slip of the tongue of Randall House, that there are 2 products on the market, and you think, yes, they are on the market, but not quite so yet, but they will be on the market, and the question only is how fast can we get them on the market.
At Duke, the primary emphasis has been how do we get the enzyme to the most people in the quickest amount of time. And I will tell you that that drives every decision that we have ever made.
Over the next 5 years, trials would continue. There would be ups and downs, but we continued to move steadily towards commercial access to enzyme replacement therapy being a reality. 2006 was another pivotal year for the Pompe community, seeing EMEA and FDA approval of Myozyme, the first ever commercial treatment for Pompe disease.
Finally, I know 15 years doesn't sound rapid, but in this type of research it is, believe me. 13 or 14 years ago, when there was no patient organization, and there was a very loose-knit group of research scientists, and successfully got them together at many conferences like this, which expanded over the years and led to a joint effort of research, competition, involvement of industry, and led us where we are today.
It was clear that part of why we had made strides was the way the Pompe community worked together. But we also saw how much more we had to learn.
I will not go into much detail, but what is important here to remember is that 68% of the patients in our study with the IVS1 mutation had this haplotype, which means that the differences in haplotypes could not explain the differences between the patients that we found.
It's been a very difficult path. It's been a very long road, very expensive road. A lot of challenges, a lot of disappointments, a lot of successes.
We needed to know more about contributing factors.
This case is a particularly interesting case because here's a brother and sister who have the exact same genotype, and yet the onset of their disease was very different. Her age of onset was 28 years old. You can see the glycogen buildup shown here. They have the same degree of residual activity, and yet his age of onset was 42 years of age. So we'd very much like to understand what these other genetic factors are which drive these differentials, and we're still trying to figure that out.
We needed to better understand the mechanics of muscle wasting.
We think that enzyme replacement therapy addresses the problem of accumulation of glycogen in swollen, expanded lysosomes, and it addresses it very successfully. But we think that it is critical at this time to look beyond the lysosomes, because it is increasingly evident that other processes, and in particular autophagy, affect the outcome of therapy.
And we were beginning to learn about the benefits of newborn screening.
I think it's very clear that newborn screening will soon be a reality. There are currently funded pilot projects, one in Washington State at the current time, and I think you can see immediately some of the complexities that will arise when you get a newborn blood spot that has an extremely low enzyme activity and deciding exactly what course you should follow, etc.
If you look at the older mice, if you treat them at 10 months of age, we can clear the glycogen to a very large extent, but a lot of the nuclei are centrally located, suggesting that perhaps one reason why we were not able to completely rescue function is because perhaps the regenerative potential of these muscles is no longer possible.
The Pompe community is not one to sit quietly by. We are always striving for better treatments, better understanding, and better advocacy. Between 2006 and the next AMDA Patient and Scientific Conference in 2011, we continue to see advances. And we also learned about exciting new developments in the field of muscle regeneration and gene therapy. And newborn screening was once again discussed.
So you can see here that in the clinical diagnosed patient, although they may be 3 or 4 months apart from the newborn screening, but there's a decrease of the survival. And half of them, even with enzyme replacement therapy, still required ventilator support.
Over the last 20 years, the AMDA has been at the center of efforts to educate, support, and advise the Pompe community. We have seen the dream become a reality. It's amazing how far we have come, but there is still work to be done. Our hopes and dreams have turned to better treatment, earlier diagnosis across the board, and access to treatment for all patients everywhere. The AMDA is excited to see where the next 20 years takes us. Beyond the scientific impact, the AMDA conferences are an opportunity for patients to connect with each other, share their stories, and to connect with the scientific community.